The overall goal of this procedure is to identify and measure changes in biofilm formation by assessing the development of wrinkled colony formation over time. This is accomplished by first growing bacterial strains of interest in liquid culture. The second step is to spot the cultures onto nutrient agar. Next, the spots are monitored over time to identify the time at which they begin to develop three-dimensional structures indicative of the initiation of wrinkled colony formation. Development of colony morphology is monitored until no further changes are visualized. Ultimately, microscopy of the spots is used to show differences in biofilm development over time by specific strains or under specific conditions in a manner that is semi-quantitative.
The main advantage of this technique over existing methods is that it permits a semi-quantitative analysis of bacterial biofilm formation using the development of wrinkled colony morphology.
Though this method can provide insight into biofilm formation in Vibrio fischeri, it can also be applied to the characterization of biofilm phenotypes in other bacterial systems, such as Bacillus subtilis, Pseudomonas aeruginosa, and Vibrio cholera.
We first had the idea for this method when we identified bacterial mutants that exhibited a delay in the development of wrinkled colony formation.
Biofilm formation is generally influenced by cell density and growth rate. Therefore, prior to assessing wrinkled colony morphology, the growth rate and yield of the strains of interest should be determined. To determine growth rate, measure the increase in optical density, or OD, of each strain over time and plot a growth curve. To determine yield, or final cell number,